Hi guys, my name is Liz Yao, and this is my sourdough starters experiment that I took part in. So first, a little background on sourdough starters. Um, they are wild yeast that is made from flour, water, and the wild yeast in the air all around us. And yeast, lactic acid bacteria, and acetic acid bacteria live in the starters. Once these bacteria are activated, the microorganisms inside them produce carbon dioxide bubbles, which make the bread rise. And so a little background on this class. I am an online student, but there are two sections. There's students in person and students online. So the students in person carried out the experiments with the sourdough starters at the lab, testing them under the different experimental conditions. And after that, DNA was extracted from those starters that they were experimenting on and processed into libraries where they were sequenced and returned. And once they returned, the students online analyzed the microbial communities that were present in the starters using the metagenomic sequencing data. And they translated the raw data from the experimental starters into the metabolic models, which we used K-base in narratives. And this is an example of what my narrative looked like. So the models were used to predict which organisms in the sourdough starters have the metabolic pathways for lactic acid production and whether they are hetero or homoformative, which that was the overall research question we were trying to address. So just some background on DNA sequencing. It determines the order of the 4 chemical bases, which are like the building blocks of DNA, and that would be adenine, thymine, guanine, and cytosine. And they all make up the DNA molecule. In this experiment, we used Illumina sequencing to sequence our data. So the organisms that were present in the original control starter were yeasts and lactic acid bacteria. This is just a picture of the lactic acid bacteria. And some of the most abundant bacterial species and fungal species were the Lactobacilli and the Lactobacilliaceae and Saccharomyces. Okay. So in K-base for the online students, we used phylogenetics to identify the species through the Kaju graphs, which is this right here. So for the control, this is the control sample, this first one right here. The most abundant species was the Lactobacillus brevis, which is this light green color right here. But the MAG that was found in the species tree for the control was the Lactobacillus fermentum. That's right here. As you can see, like the control is right here, the fermentum's right here, so it's the most abundant MAG. However, in the Kaju, the fermentum was not present, and the fermentum is this pink color. It's present. These are the low salts and high salts, but this is the control, and the fermentum was not present in the control. So could have been because it was lower abundance, but comparing that to, oopsie, let's go back. Okay. This doesn't necessarily mean that the analysis is wrong. It was just probably due to an error in the transformation of the data, or the species tree identifies the most abundant species, but sometimes in experiments there's human error inputting in the data, so it doesn't always show that way in the species graph rather than the species tree. So Okay. So here's the original data that was taken on day 2 of the experiment. And the community, if you compare this to this control right here and the other ones with the salts, the community changed a good amount. As you can see, Lactobacillus brevis is still the highest in the Kaju graph right here, the greens. However, there are different in the species tree. There's no Lactobacillus fermentum. So that changed, and that was just during a short period of growth and addition to the presence of the salt. As you can see, there is more bacteria that's present in the salts, but they still have the most abundant as the brevis. Okay, and the composition of the high salt, back to this graph here, changed more in a short period of time than the low salt, and changed more in prolonged growth. So, oh my goodness, sorry. So this is the high salt right here, and this is the low salt. This one changed more, or wait, sorry, this is the high salt, this is the low salt. This one changed more as like it has more abundance of each of the colonies, bacterial colonies. And as you can see, the low salt and the control, they have the same highest mag in the species tree. However, the high salt, the highest mag there was where is it? I believe it was the crispus. But yeah, so it differed the most from the other ones. There were changes in the composition when the low salt was added, because you can see the Lactobacillus fermentum is present here rather than the control. However, the high salt changed the most. Yeah, so parts of our original research question that were that weren't addressed. We were only able to evaluate 2 salt conditions, high salt and low salt, but we could have evaluated different levels of salt, like really low salt or really high salt, seeing if there is that what that checkpoint is to make this Lactobacillus fermentum show up. If there's a certain checkpoint of an amount of salt, and like how that works in the species tree and graph. But or we could have done another substance in the sourdough. Our research question was just to see how it affects the sourdough starter. So the most surprising result in this experiment were those when the mag species tree and the kaiju did not line up in the control starter right here, because there's no presence of the fermentum. And we could follow up on this by retrying the control starter under the same conditions, determining if we could get the same result. And if the same result is, however, if the same result is achieved, and we don't get the fermentum still, then there could be another, it's rather than human error, the error could be that the presence of the salt could be something that the presence of the salt adds to the starter, that can be further evaluated by lessening the concentration of the salt until that fermentum is no longer present, and making that the kind of like barrier or checkpoint of that fermentum showing up, if we wanted to achieve that analysis. Yeah, but this is what we did. Some of the results were a little confusing, but overall,